Hey everybody, we are going to jump into chromosomal abnormalities, which is our final set of notes for the cell cycle unit. And that means that the exam is on deck. It's coming soon. It's actually going to be next Monday and Tuesday, so a week from today. So not a very long set of notes though here, 12, 13 slides, something like that. Um, so a couple questions. How can genetic information be altered, and how can these changes lead to changes in the traits of an organism? So we're going to investigate some of those things. So the first thing we need to talk about is something called non-disjunction. So non-disjunction is when the chromosomes are lined up at metaphase, and then they do not get separated properly at anaphase. They actually get the numbers of them get shifted a little bit, and they end up with irregular numbers of chromosomes um, being pulled to the side. So if you can look down here, right, they should get pulled equally, right? When you have things lined up, they should get pulled equally. So if, this is your cell, then they should get pulled like this, where you have this one and this one and this one and this one. And then down here, you have your haploid numbers, your N, right? Well, in this case, all of this one got pulled over to the side. It didn't split. And so now this one ends up N plus one, and this one ends up N minus one, where, this guy is missing one and this guy got an extra, right? So you can look over here and see some of the same things going on. This one's a plus one, this one's a plus one, this one's a minus one, this one's a minus one. So not very many of these gametes got the proper number of chromosomes. It was just these two that ended up with the proper number. So that's going to give you some problems, okay? So um, it's really going to only affect the entire organism if it's happening in that germline cell, right? Because if it happens in a germline cell, that's this cell, right? That's up here. Okay, and these are the cells that go through meiosis. Remember, germline cells are found in which is this one and this one. They're found in the linings of the ovaries and the testes, right? And so they go through the meiosis process, and you end up with your gametes down here. These are your egg and your sperm. Remember, egg and sperm do not go through meiosis. Germline cells do. So why will this only have an effect if it happens in a germline cell? Well, that's because that's the cells that are going to be the gametes that then get fertilized, and it will be that way in the first cell, in the zygote, in that first fertilized cell. It'll be that way. And then when things get copied in mitosis, now every cell in that organism will be messed up. If it happens in just a regular somatic cell, if this happens in a skin cell, okay, that's one cell out of trillions, right? That's probably not going to affect the organism. But if it happens early on in the process in meiosis, and then every cell becomes that way, now you're talking about a much bigger issue. Couple types of rearrangements here. You have the deletions up here. So in this case, A, B, C, D, E, F, G, H. In this case, A, B, C, E, we lost the D, right? The D got taken out. So that's a deletion. You lost part of your chromosome. A duplication, parts got duplicated, right? A, B, C, B, C. Now we have more DNA than what we want. That's not great either. An inversion, A, B, C, D versus A, D, C, B. Now it got flipped and it got rearranged. So all of these things are going to rearrange the bases of the DNA, right? It's going to rearrange the ACGs and Ts, which then give you a different sequence for your mRNA, which then make different proteins, which may or may not do what they're supposed to do. Oftentimes they will not, okay? And then you have this one down here, translocation, where you basically have pieces of chromosomes kind of getting tangled and trading or breaking off and reattaching. So here you have A and B going onto this chromosome down here, and you have M, N, and O coming up to this one. And that's called a translocation, where they're actually kind of trading segments from one chromosome to a non-homologous chromosome to a different one, right? So these are all going to affect the DNA level, which affects the protein level and affects how things get done. So going into some more particular uh, problems here, chromosome problems. So single chromosome or like SOMI mutations, it's just involve a missing or extra single chromosome. Usually result in a miscarriage. Okay, so a lot of times um, you may have heard, you may know somebody that's had a miscarriage, right? And so generally, it's a super sad process, right? When someone has a miscarriage, it's not what you want to happen. People are pregnant, they think they're going to have a, nice, healthy baby, and then, there's a miscarriage, which is basically a spontaneous abortion of the zygote and of those cells that have developed because something's not right. And so while the miscarriage is really sad, a lot of times miscarriages happen because the chromosomes are not right. There's all kinds of genetic problems going on in there, and the organism, the offspring, the baby, if you will, would not have survived. It wouldn't have made it very well, or if it would have, it probably wouldn't have made it very long. It would have had a lot of problems. Okay, so while it's not, what you want to have happen, they do for the most part happen for a reason. So uh, then the question becomes, well, then why is Down syndrome tolerated, right? So let's talk about Down syndrome real quick. Uh, and its, other name here is trisomy 21. And you can probably figure out why Down syndrome is called trisomy 21 if you look up here at the karyotype. You can look through all these chromosomes. And there's an arrow sitting right here, right at the number 21 chromosome. There is 1, 2, 3 number 21 chromosomes. Okay, so trisomy 21. So an extra number 21 chromosome. And some of the traits you get are the wide rounded face, you get the slanted eyelids, the enlarged tongue. If you've ever spoken with someone who has Downs, you notice their speech is a little bit different. It can be, a little bit harder to understand. They generally have a lower IQ, but they are also some of the happiest people you'll ever meet. They are generally very happy. Um, I interact almost daily with some kids in our school that have Down syndrome, and they rarely have a bad day. They're pretty happy with who they are and what they're doing. So it's a great trait to have, actually. And so going back to this question, why is Down syndrome tolerated? Why do you think that is? Why would trisomy 21 be tolerated when other things result in a miscarriage, right? Well, the answer for this one is that number 21 chromosome is the smallest. It's super small. So there's not very many genes on it. And the genes that are on it are not controlling major processes. It's not controlling the formation of your heart. Okay, it's not controlling these major organs and things that you have to have. So trisomy 21 does get tolerated, and we do have people with Down syndrome. There's another one running down the track. So here's a little chart to kind of look at what's going on with the chances of getting Down syndrome, and really the chance of trisomy 21 occurring increases with the mother's age. So if we look at this part, so females are born with all of their germline cells suspended at prophase 1 of meiosis. The longer these cells remain at this stage, the greater the chance for non-disjunction occurring when the eggs are formed. So when you're 20, it's about a 1 in 1925 chance. 25 is about 1 in 1200. 30 is about 1 in almost 900. Okay. 35, 1 in 365. A 40-year-old has a chance of about 1 in 100 for having an offspring with Down syndrome. So your chances keep increasing the longer your cells are suspended in this area, because non-disjunction will occur at a higher rate the longer the cells are suspended at that phase. And so not that this means in any way, shape, or form you girls watching this should run out and have babies early, but waiting till you're 40, 45, 50 might not be the best plan either. Okay. So rolling into a few other ones here, a few other SOMI mutations, they actually happen with the sex chromosomes as well, the X and the Y. They're generally more accepted, fewer miscarriages. This can happen quite a bit. So Turner syndrome, or what we call monosomy X, monosomy meaning one X. So this person has an X chromosome, but it doesn't have a second X, and it doesn't have a Y. It just has one X. And so this happens about 1 in every 6,000 births. The person is still considered female. You have broad shoulders, usually a webbed neck. You can see the webbing up here, okay, in the upper part of the neck. They are sterile. They are not able to produce children, and they do not go through puberty. So a couple facts there about someone with Turner syndrome. And there's their karyotype, and you can see there's only one X down there. And here's what an adult might look like that has Turner syndrome. Okay, you can see a lot of the features do not fully develop because of no puberty. So Klinefelter's is phenotypically male. In this case, you got an XXY. You actually have 3 sex chromosomes. XX typically female, XY typically male. So that can be a little bit different as well. So 1 in every 1,500, phenotypically male because they have the Y. They are producing testosterone and they do have male sex organs. And so they are sterile. They're not allowed to have, they are not able to have children. And they do sometimes have excess breast tissue. So while they are male, sometimes they do have X breast tissue, more typically seen in females. And you can see the karyotype down here where there's 2 X's and a Y. Jacob syndrome. In this case, it's XYY. 1 in every 1,100 births, phenotypically male, taller than average, persistent acne, and once thought to be more criminally active, which is kind of, silly to think about. They thought this because with having 2 Y chromosomes, they thought that there would be, way more testosterone and some people with Jacob syndrome were at one time, they happened to, several people got arrested at one time for crimes and they all had Jacob syndrome, and people made the leap that, oh, they must have all this testosterone and they're super manly, but they're also super aggressive and they're out there committing crimes. And so there's this link here, and really there's not much of a link there at all, but that's what people thought a while ago. Multiple X and Y chromosomes. So why is this not a problem? Why is this accepted by the body? Why are they still able to develop? Well, the Y is the smallest chromosome. It has the fewest genes. Sorry, guys. And the main function is in testes development. If you have a Y chromosome, the genes on the Y chromosome are just the guy genes. They're just there to make you a guy. Okay, so sex organs, testosterone, all that stuff that make you male is what's going on in the Y chromosome. An X chromosome, while it is large and it has many genes, male and female need the X. There are things on the X chromosome that, yes, while they do have some things to do with being a female, there's also things on there that just have to do with being human, and all humans need that chromosome. It is interesting to note, though, that not every X actually becomes active. So you only want one of these X chromosomes active. So ladies, you have XX, right? But both of your X chromosomes are not really active. One of them kind of gets wrapped up in a protein that kind of coats it so that it can't make mRNA and it can't produce proteins, because otherwise you would have double the amount of proteins, and then you would have all this stuff on, hyperactive. All of your genes would have produced double the protein for the X chromosome, and that wouldn't be good either. So they get converted and they get kind of wrapped up and they become these bar bodies, is what we call them, bar, because after the scientists that discovered this. And so really only one X remains active. And if we go back and you look at someone with Klinefelter's with XXY, they would have one bar body, so that it would wrap up that X there. The Turners would not have any bar bodies. That one X, because they only have the one, there would be no other one to wrap up. So they wouldn't have any bar bodies. This person would have one bar body. Okay, so it's always one less than the number of X's. Guys typically have no bar bodies, right? Because we need our X to work. So that's that. Something called whole sets or ploidy mutations, where you actually get an entire extra set of homologous chromosomes. It's not usually tolerated. We humans are diploid, but we don't do very well. If things are triploid or tetraploid, those just aren't going to work. They're not going to form. The animal will not actually become an organism. If that were to help, it would be, aborted at the beginning of the process. Now, some plants can actually do this. Strawberries are octoploidy. That means 8. They have 8 sets. Okay, so certain plants have multiple sets of chromosomes, and I think there are other organisms, other examples of that. But I know strawberries are octoploidy, which is kind of crazy to think. A few more things about chromosomal rearrangement. There's no rule how these alterations can affect organisms. Sometimes detrimental, sometimes silent. So sometimes they don't do anything. But there are a couple I want to point out here. Here is a translocation between number 22 and number 9. So if you look here, we have 22 as the purple one, and 9 is the kind of gray one up here, and it's much longer. And here they have traded these pieces right here on the ends. So there's a little purple end from the 22 on the 9. There's a little gray end on the, sorry, that one was on the 9 from the 22. This one is on the 22 from the 9. And so they've traded these pieces of DNA. They've been translocated, right? We learned that term a few slides ago. And in particular, these are called the Philadelphia chromosome, mainly because the researchers that there were 2 researchers that figured this out. Both happened to be at universities in Philadelphia, so they gave a little homage to the town they were in where their universities were. So this one can lead to CML leukemia along with other things as well. There's one in your mitosis lab. There's a question that revolves around this process right here, so be looking for that. Here's some other translocations over here. It's a little bit complicated to follow, but basically, if chromosome 1 has a translocation with like number 11, then sometimes we get schizophrenia. If you follow this yellow pathway right here, you get schizophrenia. There's some lymphomas on here. There's some leukemias on here. If you follow these lines and which ones somehow get traded, there's lots of leukemias and lymphomas and sarcomas. Those are cancers. Okay, all of those are cancers. Here's Down syndrome. Okay, down in this one. So a lot of things that you hear about can be caused by some of these chromosomal rearrangements. And so they can lead to some pretty bad things. And that's it for today, or for that set of notes. And again, it's your last set of notes. So make sure you're getting ready for that exam. It's going to be next Monday, Tuesday. Okay. Hope that was helpful, and let me know if you need anything. Take care.